security architecture team. I recently took an assignment as chief of staff for Bioinformatics, which has been a lot of fun. I am very pleased to introduce a longtime friend of mine. We actually, believe it or not, met through HPN. I know you're probably looking at me sideways. How did I get in Yen? We met at HPN probably 8 years ago or so, something like that. Leo Elworth is our speaker today, and at the time he was computer science in some project on more of the enterprise side of things, and we had a lot of great intellectual talks. And then he decided to leave HP, I don't know why, and I thought it was a bad decision, but then about 6 months later he just called me up and said, hey Ken, I'm going to be a PhD student at Rice University, and he was going to be studying the genome and intersection of that with computer science. So I am extraordinarily pleased and proud as a friend to introduce Leo Elworth, who is a 5th year computer science PhD at Rice. And in about 1 month, I'm going to have to call him Dr. Leo. I'm not sure I'm going to get used to that. But he is advised by Dr. Louis Nekele. I'm horrible about my pronunciation. Who is a J.S. Abercrombie professor and chair of the Department of Computer Science at Rice. Since joining Rice, Leo was awarded a graduate research fellowship from the National Library of Medicine, has published work in computational biology in journals such as Bioinformatics, you're going to learn more about that right now, and presented research at scientific conferences like RECOM CG in Barcelona and WABI in Helsinki. Without further ado, please help me give a warm HP welcome to our speaker today, Leo.
Thank you so much for that very kind introduction. I'm absolutely thrilled to be here today talking to you all about bioinformatics. Bioinformatics. What is bioinformatics? So can I just start with maybe a quick show of hands? Has anybody ever heard this word before? Know anything about it? Okay, so we have a few people. Most have not. Well, let's go ahead and just start with a quick definition. I'm going to give you my lightning fast definition of bioinformatics. To me, bioinformatics is just the intersection of computers, computing, computation, computer science, and genetics. So DNA sequences, how you get the different traits that make you up from your DNA, or even more broadly, maybe just biology in general. Now you can imagine when you take computer science and biology or genetics and you intersect these, a lot of interesting things start to happen. One thing that happens is there's an absolute explosion in the demand for things like computer storage. Another thing is there's an explosion in demands for CPU cycles, actual computations to run the different pieces of software. There's also just a lot of really interesting stories and anecdotes that pop up in this field. So I'm going to walk you through these interesting stories. Let's start on the International Space Station. I'll just refer to it as the ISS. The ISS is, of course, floating there in space right next to us, where the astronauts are hanging out. There's a lot of interesting science that happens on the ISS. There's also some not so interesting things. They have to do maintenance, they have to change out air filters, they have to do a lot of boring things. Well, not that long ago, this was around 2014, they decided to change some air filters out. They took the air filters from the ISS, they brought them back to Earth, and they wanted to do some science experiments to see what's going on up there in space. What are the bacteria that are in the air up there in space hanging out with the astronauts? So they bring the air filters back down to Earth, and they look at the bacteria to see what's in there. The first thing that they do is a very basic test. They just try to grow the bacteria a little bit. So they'll do what's called culturing. They'll stick it in something that the bacteria like. In this case, they stuck it in sheep's blood. For some reason, bacteria really like sheep's blood. I don't know why. They grow really well in that. So they stick it in there, and the bacteria start growing. And then these things are going to grow large enough that you might even be able to just look with your human eye and see what's growing there. Or maybe you use a microscope and you look and you see, does this look like anything that we've ever seen before? Well, it did look like something that we've seen before. Actually, it looked like anthrax. So there was this scare that ran throughout the government. They were actually really worried that there was anthrax on the International Space Station. They actually went and they sent the you please look at this and tell us what it is? These other government agencies, they said this looks like anthrax. So this question pops up. Is there anthrax on the International Space Station? I can only sort of conjecture on this for various reasons, that maybe the ISS could have been actually shut down with this, or parts of it would have been shut down. We're actually very worried about this. And we needed to answer this question. So how do we answer this question? Do we just look inside of the sheep's blood and say this kind of looks like anthrax, maybe it's anthrax, I'm not really sure? Or are there other tools that we can use? Do we have better ways to definitively answer these questions that pop up? Is this anthrax or not? We do. So I'm actually going to walk you guys through how you do this. There's 2 steps. The first step is you have to sequence the DNA of the organism. So you have to take this bacteria, feed it into some kind of magical machine that tells you exactly what the DNA is inside. I'm going to go more into detail on these 2 steps in just a moment. And then finally, once you get that data back, you have to actually analyze it and figure out what can I glean from this DNA? Can I learn something from this? Can I analyze this and actually answer my question? And of course, everybody likes talking about big data. Well, in my opinion, this is about as big data gets. Maybe at some places, I mean, you guys obviously have a lot of data with customers and things, and social networking sites maybe, but this has to be absolutely one of the biggest. These DNA sequences are extremely long, tons and tons of data generated constantly. So how do we handle all of this? Well, before I answer this question, is there anthrax on the International Space Station? Let's start by answering the question, who am I? Up here telling you about these things. Well, let's just start at birth and work our way from there. So I'm actually from a very small town in Willard, Missouri, way out in the middle of nowhere. The population was around 2,000 people whenever I was born there. It was mostly just animals walking around and a lot of trees and things like that. So when I was born, the first words coming out of my mouth, unfortunately, were not computer science and genetics and bioinformatics. They weren't even mom and dad. The first words that I said were cow and moo. This was how I started out in life. There's me saying these things, maybe. Apparently, I would sit by the window and look out and just moo, moo, cow. Well, very quickly I learned I don't really care that much about cows and trees and things. I'm more interested in computers. One of the things that actually really got me started down this path was video games. I absolutely love video games. All growing up, absolutely love them. I still enjoy them. I don't have quite as much time to play them as I like anymore. The first video game console I ever had was a Sega Genesis. I bet a few people have heard of this before, at least one person. There was a game called Sonic the Hedgehog on it. Thank you. And I just loved it. And I had to know, how does this work? How do you make this? And I really wanted to make video games and understand how it all worked. And so that actually was what led me to decide I wanted to learn more about computers. I wanted to learn how these things worked. I wanted to make video games. And I decided to go and get a bachelor's in computer science at University of Arkansas. Immediately following that, I actually came here and I worked as a software engineer here at Hewlett-Packard, which was a very nice experience. I got really involved in a lot of different organizations, which I'm going to tell you a little bit more about a few that I really think you should all look into in a second. Then after that, I decided to go for a PhD in computer science at Rice University. So I've been there for about the last 5 years now, trying to graduate in the next couple months. And then actually immediately after this, I'm going to stay as a postdoctoral research there for at least another year or two. I'm joining the lab that I'm actually about to join next year. The head of the lab, his name is Dr. Triangan. He's actually the one that the story I'm telling you. When the government went through all these agencies and they were asking, is this anthrax? He was actually the one that had to make the final call in this particular case. So I'm going to work with him starting next year. Before we get back to the anthrax, this is what I looked like back in the HP days. So all of these pictures, actually when I went to go find some pictures of myself in this time, all I have are pictures from events from this organization called the Young Employee Network. I don't know if maybe some of you have heard about this, some of you are maybe in the Young Employee Network. I absolutely recommend, if you're a newer employee, you can check it out and go to them. The networking obviously is amazing. I had a really good time at all the events. One of the people in one of the pictures on the top left is sitting right there. Thank you, Ron. So just last weekend, I was hanging out with people that I met through Yen. Really amazing organization. Another amazing organization that I have to call out for my time here at HP was the Houston HP Toastmasters group. It's a public speaking group here on campus. You get to practice, get better at all these things. Ken actually introduced me to it, and it's been one of the best experiences I've ever had from a professional standpoint, I would say. So they have that here. Absolutely The DNA is essentially the instruction booklet for who you're going to become and different traits that you have, like your eye color, things like that. Maybe even how tall you're going to be. Obviously, the environment plays a role as well, but coded in the DNA are all these different instructions to make you who you are. Not only to make you who you are, but also for bacteria to make them who they are. In fact, all living organisms on planet Earth have these DNA molecules that determine who they are, what they're going to become, and the different traits that they have. These DNA molecules, they're just a really long molecule that has these repeating patterns over and over and over again. So as you can kind of see in this graphic here, you have this one long molecule with a whole bunch of little molecules that repeat over and over and over again. We just refer to these as T, G, A, and C. And they just repeat over and over and over again in this long chain. The pattern that they form in this long chain is what determines things like your eye color, etc. So if you can actually get in and look and see what's in there and actually find the exact letters that make up the DNA in your cell or in the cell of bacteria, you can learn all these things about the organism that you're looking at. In humans, this really, really long sequence of these letters is about 3 billion letters long. There's different words that are used for these things. You can call these things base pairs, these T, A, G, C molecules, you can call base pairs. I'll probably call the letters of the genome. You can call them nucleotides. So the actual term, of course, DNA is deoxyribonucleic acid. These are technically called nucleotides, but all these can be used interchangeably. It's no problem. So in humans, to actually sequence the DNA of a human is to go into the cell, get the DNA out, and actually figure out all of these consecutive letters. And you can have a file on your computer that just says all of the letters inside of your DNA consecutively. For anthrax, you can do the same thing. Although their DNA is a little bit smaller than ours, it's only 5 million letters instead of 3 billion letters. And we have the technology today to actually do this process where you can get the DNA out of your cells or out of bacterial cells, and you can figure out exactly what these letters are. Here's just one very quick example. This is kind of a newer technology. It's from a company called Oxford Nanopore. They've made these devices where you feed in the DNA from your sample. So maybe I take some blood from somebody, or maybe some saliva from a human. I feed it into this machine, and the DNA, inside this machine there'll be these just absolutely tiny things called nanopores, and the DNA will just feed through this nanopore, and as it's being fed through, the nanopore will detect what these letters are as they're going through. And there'll be this little electric current that will be changing as these nucleotides are passing through, and you can see how it kind of goes up and down as the letters pass one by one through this. And that tells you if it's an A, a C, a T, or a G. And once all of the DNA has passed through, you have the full DNA sequence to be able to actually analyze and figure out what's going on there. Well, what do you do then? If I just hand you a DNA sequence, what do you do with it? How do you figure something interesting out about it? You have to perform some kind of analysis and figure out how to glean the actual information out of it. So let's say I was going to try to sequence somebody in this room, and somebody handed me some human DNA, but I didn't know it was from a human, and I had to figure out, where did this come from? Well, before we do that, we already have a bunch of DNA sequences that we've done this process for. We've sequenced humans and chimpanzees and gorilla and mice, lots of bacteria. We've already done a lot of sequencing up to this point. So we have these databases where you can just go look up what all these sequences look like. And when you line them all up, so let's say we take a human, a chimp, a gorilla, a mouse, and we just stack their DNA sequences on top of each other, most of the DNA looks identical. Every single site, as you go from left to right, just looks exactly the same between all these different creatures. But of course, there's some differences. Obviously, we're different than mice and chimps, so we have our own unique mutations. So most of these letters are going to look like, in the gray, these ancient variants, things that have been passed down to all of us through time that we all still have. That's what most of the DNA looks like, but it doesn't all look like that. So you see, in the blue, there might be some mutations where these letters change over time, where the common ancestor of humans and chimpanzees, at some point there was a mutation that affected one of these sites, one of these letters, and ourselves and chimpanzees still share this commonality in this particular letter. Of course, humans, we're going to have our own mutations that define what it means to be a human, that makes us who we are, gives us big brains and other things. So now let's say I pick some random person. Let's say to what we know to be human DNA. And we can therefore figure out, without you having told me, what this came from. It came from a human. We know these mutations, that we know that they're specific to humans. We've already sequenced a bunch of humans, so now I can match these and I can figure this out. It's not from a chimp that has that A there. It's not from a gorilla that has that A there. This is from a human. Now we've answered this question. Well, it works exactly the same with bacteria. So if I just look at the bacterial case, the letters are going to look a little bit different. So we'll have something like this. But still, if you hand me a whole bunch of different bacteria, so let's say we have E. coli, a very common bacteria, and let's say I have its DNA sequenced. Let's say I have some other random bacteria and I have its DNA sequence, and I'm stacking all these on top and I'm comparing all of them. Well, we've sequenced before Bacillus anthracis. This is the bacteria that causes anthrax. We've also sequenced something called Bacillus cereus. This bacteria does not cause anthrax, but it is extremely closely related to the anthrax bacteria. Only a few mutations actually separate these two, but these mutations that separate them are crucial, and this is actually what causes the disease in humans. So now, if we take these air filters from the International Space Station, we're going to feed the bacteria into these DNA sequencing machines. We're going to get the full DNA sequence. We're going to stack it with all these others, and we're going to compare it to all of the sequences that we know. And we're going to find the same thing what I just described to you. We're going to find that all across, when it has these unique mutations, they all match with this Bacillus cereus bacteria. Therefore, it is actually not anthrax. So this was the final determination that was made. I'm trying to keep this talk kind of high level, but I am going to go kind of in the weeds a couple times. This image right here on the right, this is the actual software that you use to do these things. You know, the scientific computing software for stacking all of these sequences. And this was one of the main pieces of evidence supplied to the government to show them this is not anthrax. It's okay. Don't worry. It's not anthrax. Very closely related, but not. So Dr. Todd Trank, who I'm working with next year, he actually generated this. What you see here on the top is a whole bunch of sequences that were sequenced that we know to be Bacillus cereus, this close relative bacteria. And then on the bottom stack is all of these DNA sequences from Bacillus anthracis, which causes anthrax. And then they sequence the full genome, the full DNA sequence of the bacteria from the International Space Station. And you can see there's this one particular site that tells us this is Bacillus cereus, it is not Bacillus anthracis, this is not anthrax. And we can all take a deep breath and not have to worry too much about this anymore. Okay. So that wraps up this question. We have an answer, and this is how we get to that answer. This is also how we answer many, many questions with genetics. You have to sequence the DNA, and then you try to answer your question afterwards. And as I said, there's a lot of interesting stories and anecdotes you get from this field, so let's go ahead and just jump into another one. This time, I bet a lot of you will have heard of this story. I bet a lot of you remember it. This was right after September 11th. There were these letters that were going around in the mail. It was very terrifying, and there was anthrax inside these letters, so we're still going to stick with anthrax here. The letters look kind of like this. This story, I mean, really to me, it is kind of a chilling story. People were dying. Mostly the people that died were actually in the mail service. The targets were like Congressman and Tom Brokaw, people like that. People were actually dying, and you read this, and to me, this is just really terrifying. You cannot stop us. We have this anthrax. You die now. Are you afraid? Death to America. So these were the letters that were being mailed out, and there was just like powdered anthrax inside of these. You open it up, and then you most likely will die from that. So we have another question here. So the question this time is a little bit different. The question is not, is this anthrax or not? We know that it is. The question this time is, can we figure anything out about the anthrax? Can we maybe, for instance, trace it back to where it came from? Well, it turns out that you do it in exactly the same way. So step one, the first thing that they had to do was they had to sequence the full DNA of all of the anthrax that were in these letters. And then we looked to see, do we know anything about, for instance, the labs around the country? We'll have to go sequence the DNA of the anthrax in these different labs, and we'll see if we can find a connection, if we can find some mutation that links it back to somewhere in the United States. They were thinking that this was probably a terrorist. It seemed like the two were connected from 9/11 and this also. And so let's dive in once again into the weeds a little bit here, and let's actually look at directly from the scientific paper that was published as a result of this. The people that looked into this and answered this question, they published this paper in the Proceedings of National Academy of Sciences, and this is the claim that they made, which ties all this together. It ends up being the exact same thing. simple as that. Unfortunately, in real life, it's not that easy. It's actually a little more complex than that. And this is where it starts getting more computational and where we start getting more and more data storage requirements. This is what it actually kind of looks like. You stick the DNA into the machine. The first thing that happens is you have to make a whole bunch of copies of the DNA. So let's say I'm only feeding in like 100 cells from a human. That's 100 copies of the DNA, one in each of the cells. The first thing that happens is they might make like 1 trillion copies of it. So they amplify the DNA up like crazy. You got, let's say, 1 trillion copies, and then you mash all of it up and chop it up into these teeny little fragments, so that when the machine reads the DNA, it actually only reads it in teeny little fragments. So the machine is going to give you one fragment of maybe 100 base pairs, and then it'll give you another fragment of like 100 base pairs, and another fragment of about 100 base pairs. And what you end up with is this file on your computer that might have 1 trillion of these really small little miniature reads that are only a piece of the full genome. And then what you have to do is you actually have to stitch them all back together to actually get the full DNA sequence. What you usually have is, as shown here, this reference. Usually someone else will have already sequenced the genome of the organism. So let's go back to sequencing a human being. We've sequenced a human being, so we know in general what a human DNA strand looks like, what the full sequence looks like. Now, if you're getting these 100 base pair reads out of the machine from someone else in the audience, you can just map these directly onto that sequence that we already know. And then you can just find, maybe there's a few places where it differs. So for instance here, that red gene, we've mapped all of these new reads onto the original sequence that we know, and you'll see like all the reads have a gene at that site, so that particular person maybe has a mutation at that particular site. And then you reconstruct their full DNA sequence from all of these teeny little fragments. There could be errors also. Some of these reads are incorrect. Not only that, the machines, when they generate these reads, for every single 100 base pairs they generate, they're going to add all of this extra data from the machine. They might say, I think I might have messed up on this base pair. I'm only 50% confident that I got this one right. All kinds of extra data. So now these machines are spitting out, every time you want to go do DNA sequencing for one sequence, the machine spits out all of these reads, all this extra data along with it, and the storage requirements just go absolutely out of control. Another quick metaphor for this would be something like, you want to learn about a tree in a forest, and you say, hey, can you tell me something about the tree, or can you show me what it looks like? And I say, yeah, sure, I'll show you what this looks like. So I'm going to take the tree and I'm going to feed it through a wood chipper machine, and then I'm going to hand you back all of the wood chips in the end, and I'm going to say, here's the tree, here you go, I gave it to you. And then you have to go and reconstruct everything from the wood chips. Another nice analogy is like for books. You say, can I please have that book? I say, sure. I feed it through a shredder, and then I give you little fragments of sentences, like 5 words from a sentence at a time. And I say, here's the book, and then you have to reconstruct everything back from that. So these storage requirements for doing this, to actually store this on computers, one single run of one of these machines, when they're generating all these reads and all the extra data that comes with it, one single run could be a few gigabytes of data to store. It could be up to 2 terabytes of data. So some of these machines, you try to sequence one genome for one person, you could get 2 terabytes of data back. 2 terabytes. I'm assuming most of the people have a good idea about how much that is. Yeah, I only have 1 terabyte on my laptop here that I'm using for this presentation. I have 1 terabyte. One single run for one sequence, 2 terabytes. I can't even, I'm already done on my laptop. I can't do it. So this gets out of control very quickly. There's a lot of sequencing being done right now. The cost, so let's analyze this for a little bit. What I have here that I'm showing you, this blue line that starts on the top left, this is the cost of how much it's cost to sequence one single genome. I really think that this is actually a marvel of modern human science. The first genome that we ever sequenced, it cost about $3 billion with a B. $3 billion, this was called the Human Genome Project. So we spent $3 billion to get one single genome. And look at what's happened to this price. Every year, just exponentially to the ground, the price of doing this. To where now, you can sequence a genome for around $1,000, or maybe if you get lucky, maybe a few $100. So from $3 billion in about 20 years to a couple $100 from $3 billion. Well, while that's taking place, people start using the technology, and as it keeps getting cheaper and cheaper, and if you tell me, just personally, I can sequence your genome for $100, I'm like, okay, let's do this. This sounds interesting. And I think a lot sequencing being done. It started out, of course, slow, and then this huge exponential growth, where now we're looking at like 1 billion sequences that we're going to have sequenced. And I'm telling you that each time you do this, you might be using gigabytes or even terabytes of storage to do this. I think you can imagine where I'm going with this. And this is where I think I have to issue somewhat of a call to action. What are we going to do now? Where do we go from here? We have all these sequences being generated. We have to analyze all of them. We've got 2 terabytes maybe per sequence. We're sequencing 1 billion of these, and then we're trying to compare all of them against each other. Think about the computational requirements and the storage requirements. Now, your sister company, of course, has been somewhat involved in these things. A lot of these things use servers when you're doing these different computations. When you're doing the sequencing, you're going to use servers. But we want to push these things away, possibly, from servers. So I'm going to give you one final, actually interesting story, anecdote from the world of bioinformatics. There was a paper published in Nature just in the last couple of years. And what the scientists in the paper wanted to do is they wanted to go to Africa to start doing DNA sequencing on the Ebola virus. Ebola was very scary for a while. I think we all maybe sort of forgot about it now. I think it's kind of under control, still a little bit scary. They wanted to go to Africa where it was really rampant and start doing the DNA sequencing to figure out exactly what was going on with the Ebola. Well, these sequencing machines are getting, not only are they getting cheaper, they're also getting smaller. So some of the latest sequencing devices are maybe about like this big to do the DNA sequencing, and it's a couple $100 or maybe $1,000. Well, they just loaded these things and then they got on the airplane. They just put it in your backpack and you put your laptop in your backpack, you get on the airplane and you go. You go to Africa, you set up a tiny little space with a bunch of laptops, a bunch of these little sequencing devices, and you just start sequencing, in this case, Ebola. And you can actually do this more like edge computing, if you want to call it that, on your laptops, on site, on location. They did have to still upload some of the stuff to the cloud still, so it wasn't all on laptop, but the point is we can push these things down to that level. And so what do we need to do that? Well, we're going to need better notebooks, mobile workstations, high-end desktops, and of course this is probably going to sound a little cliche, we need more storage, we need more RAM, we need more CPU, maybe a GPU in there, hopefully a very powerful one, fast networking. Actually, in the Ebola paper when they were doing this, they said when they were uploading some of the stuff to the cloud, they're in Africa trying to get these sequences sequenced, and they're uploading all their data to the cloud to do a piece of the computation, and they ended up getting bumped to 2G networking while they were in Africa trying to do this. And they were stuck when that happened. They couldn't do anything anymore. So can we find a way to push all of this more localized with better computers, with more specs, more storage, to actually do this on the edge? And then the final thing we need, is some innovation in this area. There's so many different shapes and forms I think that this could take, where the innovations could happen. I think there's room for innovation at the level of the hardware. For instance, there's been these things popping up, NPUs, TPUs, neural processing units, tensor processing units. These are some new, I'm not an expert on this piece of it, these are some new architectures forming around machine learning and AI because everybody's running these really data-intensive AI and machine learning applications. So they're actually creating dedicated hardware for those, these NPUs and TPUs. So I say, well, if there's NPUs and TPUs, then why aren't there BPUs, a bioinformatics processing unit? There's a lot of computations that are being done over and over. There are certain when you're stitching the reads together, when you're doing the sequencing, this is happening over and over again. There's these sequencing machines running in the Houston Medical Center, basically 24 hours a day, just rooms filled with these things, just going, cranking out DNA sequences. And there's these computations have to be done over and over again. Maybe there's some room for innovation there. There's always room for innovation at the level of some computer algorithm, some way to actually use this data on your computer. Maybe we can make them faster. Maybe we can find ways that instead of storing it as 2 terabytes, maybe we can shrink that down somehow, compress the data down. So there's all this room for us to figure out how we can make this better, how we can innovate and improve all of these processes to answer all of these interesting questions that we have right now. So I've told you where I think this field is generally headed: more data, more computation, maybe pushing it more towards laptops. Now I'm going to tell you, to finish off, where I'm headed next. So I have some questions and things I'd like to look into. For starters, does anybody know what the Rice mascot is, and would anybody like to say? Okay. Does anybody know what kind of owl it is? Okay, good. Yeah, he got it. Okay, so it's the great horned owl. So I plan to, along with a couple other people, publish the first ever genome sequence for the great horned owl. I'm going to do it with these guys here. This is the lab that I'm going to be joining next year. So I'm there on the left holding the owl head. We're actually shooting a little promotional video to actually ask for the money to sequence the great horned owl. Okay? The guy in the middle there, this is Dr. Todd Trangan. This is the head of the lab that I'm joining. This is the one where, as I told you in the ISS story, when the government wanted the final answer, he was actually the one who was doing the DNA sequencing, figuring out where the mutations were that he actually reported back to them on that. So I'm going to join his lab. And we plan to start by just sequencing the great horned owl. Why are we doing this? Well, we're just doing it for fun. We're just doing that for fun. That'll be a fun little project. But I'm also interested in more practical things, obviously. I'm really more interested in like human health impacts that we can have for these things. So one of the first projects I really want to start getting going next year while I'm at postdoc at Rice is actually the following. I think we should be able to use this technology to do something like the following. Whenever you have some kind of an illness, let's say you have a throat illness, it's probably some kind of a bacteria or some kind of a virus. When you go to the doctor and you say, hey, my throat hurts, they're going to say, most likely, well, you probably have the flu or something similar to that. Maybe you should go home and rest. Or maybe I'll give you an antibiotic. I don't know. Sure, why not? I'll give you an antibiotic. So I think we can do better than that. I think we can actually do a lot better, and I know that we can. So once again, what are we going to do in this case? Step one, you're going to sequence the DNA inside of your throat, so we'll get some saliva from you. So let's say you present yourself to a clinic or wherever, maybe not even you have to go to a clinic. You take some saliva from yourself, and we can sequence the DNA, and we can tell you exactly what you have. It's not going to be, well, maybe you have the flu. Maybe we can do like one test to test for like strep throat or something like that. We're just going to fully sequence the DNA and tell you exactly what you have. And then maybe even create some kind of automated report for you. Alongside that, cancer genomics, of course this is a genetic illness. It works the same. If you can sequence the DNA of different cells in your body, it can tell you something about cancer. I'd like to look into, for instance, moles, doing like screening on moles. Let's say you have a bunch of abnormal looking moles. You can take pieces of each of the moles, run it through a DNA sequencing device, and maybe be able to tell a person, okay, this particular one here is possibly precancerous, or we know that this is precancerous. We can remove it now before it actually moves on to full-fledged like skin cancer. And with that, I'd like to conclude and say thank you very much for listening, and absolutely happy to take any questions that you have.
Can we first give Leo a round of applause? Question, raise your hand, and I'll run the mic to you as fast as I can.
Hi, Jim. My question is, the nanopore technology, you're running a human genome through there. How long does it take to sequence it?
Sure. Yeah, so this is not my absolute area of expertise, but I do have some idea. I think you can get results back in about 24 hours. Yeah. In fact, when you start for these nanopores, these are really, really new. When you start on these ones, you can start, it's going to start going one at a time. Like I told you, you fragment the genome down into little pieces. So it can start giving you like the first read, basically instantaneously. You stick it in, and you're essentially getting that first read out. But if you only get one read that's 1,000 base pairs long, obviously you have to let it run for a while, and it keeps going. And you'll wait maybe around 24 hours, and at that point you might say, okay, I think I have enough. Because the question, and this is where the data gets out of control, there's not really a stopping point on these things. You don't really know at what point, because these are random reads that you're getting out. You don't know if I've gotten everything I need to fully reconstruct the full genome yet. So a lot of people, if you have a lot of money, if you're a huge institute, like a huge research institution, you just pump in $10,000 or $20,000 and you just assume, okay, surely I'll get enough at that point that I can actually reconstruct the full thing. So there's a lot of different, variables and components that go into these things, but they do start right away. When they were doing this Ebola project, right away you can start getting results, and I think their turnaround time in that paper was around 24 hours to hopefully get a mostly fully assembled genome of these things.
Kind of following on with the question just asked about how long does it take to process that data after you've gotten it out of the, after you've sequenced it?
Sure. Yeah. Yeah, so it's really going to depend on what exactly you're trying to do. So what I was showing in the slides, I was showing you're comparing it to all these other genomes. It's going to depend on how many you want to compare to. So let's say I want to compare my DNA against your DNA. I have to sequence us both, and then I have to lay them out essentially from end to end. It's a little more complicated because in human cells, it's not one long molecule, it's 23 molecules. You have the 23 chromosomes. This is where 23andMe gets its name from. So we would take each of our chromosomes, or if you want to think of it as just one molecule, that's fine too. And we would stretch them out from end to end, and we would stack them up against each other, and then maybe look at each site to see if they're the same or not, let's say between me and you. I would say for 2 human genomes, for the 2 of us, pretty much instantaneously. But the problem is, once you get to about 10, that you want to do all of them at the same time and compare all 10, then all of a sudden you start running into some problems. So if you want to do, let's say, 100 genomes, and you want to line them all up and compare all of them against each other, then I think you're talking more on the orders of like months, and you are going to be pushing these things onto supercomputers. Yeah.
Who's next?
I'm wondering how easy or difficult for Jim to become human.
Very difficult, I would say. Impossible. I'm just going to say impossible. Yeah, I mean, it would take however long it's taken for us to diverge, it would take essentially at least that long. So I'm just going to say impossible. Yeah. Or you mean like maybe gene edit? Like gene edit?
Gene edit, yes, that's what I was
Is that what you're thinking? So there may be possible. There may be possible. Actually, I just was listening to a podcast just a few days ago where one of the leaders in all of the field of bioinformatics was on this podcast, and they were talking about resurrecting these extinct animals. I don't know if you've heard anything about this. You can take an animal right now, and if we can actually sequence the DNA from something that's gone extinct, so for instance, woolly mammoths was one of the examples they were using. If you have some kind of like ancient woolly mammoth specimen, like some bones, you can scrape out old DNA from that and actually sequence to see what the DNA looked like back then. And then you can actually find all the places, say take a modern current elephant, stack the 2 up against each other, find all the places where they differ, and then edit it back to being a woolly mammoth, let's say. So in that case, more possible, if you wanted something like that. Yeah.
With all the work you've done looking at human DNA and things like that, how much is common among all people that it's the same always? And if it's the same always, can you remove that when you're sequencing it to speed your process?
Yeah, absolutely. Absolutely. I think this is going to be one of the main active areas right now in shrinking this data down. So humans, we're like 99.99, some number of nines, I believe, identical to each other. So it's going to be very rare. There's going to be these huge stretches that are just identical everywhere. I think when you actually do the sequencing, when you're actually figuring out what your DNA looks like, I can't make any assumptions necessarily of which sites you're going to have different than let's say, some reference, or if I'm using my DNA versus yours. I won't know ahead of time which ones I could, let's say, ignore. But then once I've done the process where I've mapped all the reads onto mine, or some reference genome that we're using, and I figure out which ones are the ones that are unique to you, then I think absolutely, I think you can do exactly what you're saying. I think you can just store your differences versus some reference that we have instead of store everything. But right now, a lot of people, here's another thing that's happening. When you run these sequencing machines and you get, let's say, 2 terabytes of these reads, we're not 100% certain that we've perfectly processed all of that, because there's still a lot of mistakes that are being made just in the algorithms and computer code that they're using to do mapping. So a lot of people want to keep a hold of these so that if we figure out better ways of stitching them together, we might actually go back and redo your genome later and reconstruct it, because we might have made some mistakes right now. So I think kind of both. I think on the one end, if we're totally sure we've gotten yours exactly right, I think we can shrink it down and only store a few of the differences. But then if we think we're making some mistakes now, then I think we hold on to it at least for a little while longer.
Hi there, sorry. So how do you keep this ethical? As costs are reduced and it becomes more accessible, like what are the plans? I'm assuming there's people at the top that are worried about this, and when it comes to data protection, this is a huge deal.
Absolutely. Absolutely. I'm not as interested in that particular. So I want to just somebody to just hand me something I can just do anything I want with and not have to worry. Obviously, because I'm selfish in this way, because it's kind of annoying to deal with these kind of things. So I'm not going to be personally looking into that, but absolutely there are people looking into this right now. And yeah, you have to. I mean, for human DNA, if you ever work with human DNA, you do have to be careful with these things, and you have to make sure that you get the proper permissions, and you're keeping everything legal, and that you're keeping everything secure. So this does have to be dealt with. I think, this would be one that I think I would push back to you guys, honestly, in this room. I think this is another one we can easily add to the call to action, is the security component. How should we be storing these things? Where should you put it on a computer? How do we keep it locked down? Let's say you just sequence your DNA yourself, and let's say 23andMe says, okay, I'm gonna go ahead and just give you the files of your own DNA. Good luck with this, store it however you want. And then I think this is going to be pushed back again more in this direction here. How are we going to put these things on the laptop? How are you going to keep it secure? But absolutely, it's a huge concern. And once again, this is another area that people are very actively focused on right now, I would say.
Mine is going to be a pretty basic one. You probably think I failed biology. But, how stable is your genome? I guess if I look at it on my toe versus my hair, is it the same?
Yeah.
And then how stable is it over time?
Yep, sure. Yeah. I really like this question, actually. Yeah, it's not stable across your body. So this is the whole thing about cancer, is as the cells are duplicating, they're making mistakes. So every time you get a cell replication, there's going to be a few mistakes that are made. It's been calculated, I don't know the exact number, but there's some rate that we assume. When you have one cell division, we assume a few mistakes are going to be made. Your DNA is 3 billion letters long. Let's say every time you get one cell division, let's say like maybe 100 of those are probably going to be incorrect whenever it gets copied from one to the other. So as your body is being formed and these cells keep dividing and replicating, you're accumulating these mutations all over the body. And this is why I'm saying, like with the moles, a lot of these, maybe we could go look inside and we can just check and see what are the mistakes that have been made in these different areas. Because some of these mistakes, there are certain areas of the DNA that have been shown, absolutely, for breast cancer, for instance. There's been a ton of breast cancer studies been done. They found that there are very, very specific areas inside the genome that if a few mistakes are made in that region, probably you're going to end up having breast cancer, or it's very closely linked. So if we can find these things in your body, then yeah, maybe we can help. And yeah, you do have these all throughout. And so it's going to be the same thing. Like when you're having your child, there's going to be a few mistakes that are going to happen in that process that get passed on. There's some rates that have been figured out. You can say maybe 1 out of every million base pairs, let's say, will get changed in each generation, and that's how these things start accumulating over time.
As soon as you start to get more data and this starts to become more of a heavy topic in the field, how soon do you think predictive analysis and AI are going to start to be incorporated?
Well, I would say now, honestly. I think now is the time, and I think when I'm saying these things about, I'd like to do this, I'd like to do automated health reports, I'd like to look at precancerous moles, I'm not like a super genius with these things. There's a ton of people that are already doing these things. There's the signs are on the walls, like it's been written, you can see where things are headed right now. So AI and machine learning, they're being applied right now. And everybody right now is the time really to be in this area and to be doing these things. So, for instance, there's a company, Illumina. I mentioned Oxford Nanopore does some of this DNA sequencing. There's another company, Illumina, that does DNA sequencing. Internally at Illumina, they have people looking at these things. They're trying to figure out things like, how are we going to determine if somebody has cancer or not once we do their DNA sequencing? And you can use some machine learning techniques and artificial intelligence techniques to actually figure out this is the thing that will tell us if the person has cancer or not. Yeah, so the answer is right now. And people are doing it, and I think it's going to keep growing. I think more and more. So I'm going to say right now.
Hi, maybe possibly related. Would you recommend that the average person here use a service like 23andMe? What is the value of the layman having the files on their computer, the DNA sequence?
Yeah.
Or have you done it?
I still haven't done 23andMe. need to. I'm just cheap. Actually, I'm gonna see if I can get it done for free. So. I have looked at the 23andMe stuff. I've looked at the reporting that they do, and I don't know exactly internally how they're doing it. I know what I've learned in this area personally. I know all the research that I've read on these things. Everything is going to be proprietary, but I assume they're probably getting it right. So when they have all these reports that tell you maybe you're at risk for this particular disease, or maybe you should get this checked out, I think that it's pretty fair to assume that they probably are getting this right. So I would say, yeah, you should probably get it done. Honestly, this 23andMe stuff, and from what I understand, they're actually adding more and more every day. Going back to the AI machine learning conference, they're doing the same thing in 23andMe. They're collecting all this data from all these people. They also ask everybody that they get the DNA from, they ask you, have you ever had any of these diseases? Has this ever happened to you? And then they use the machine learning to try to figure out, aha, we've gathered enough data, we figured out all these people have this in common. So I definitely recommend looking into these things. I think it's going to be very useful for everyone. As far as if they say, well, now we've given you this report for you to use and you understand more about yourself, as far as if they say, I'm going to just go ahead and give you the files to have on your computer, your own DNA, I don't know how useful that's really going to be right now, because again, you have to know what do I do with this genome file now on my computer. I don't think there's much out there. Maybe as more public, maybe there'll be like publicly available websites that will help. They'll say, we'll do more reports on you if you upload your DNA data to us after you've gotten it back from, say, 23andMe. I could see that being one area in the future. I don't think it's really happening right now. But overall, I think very, very useful. And possibly having it yourself might be useful, we'll see.
So just a quick question, what do you think the future of human gene editing is?
Oh, man. You guys are asking great questions. Thank you.
Like 10 years out.
And making my life very difficult with some of these questions.
Like 10 years out,
Yeah.
20 years out.
The future of human genome editing. Yeah, there was actually, I don't know if anybody, so has anybody heard of the CRISPR baby? Thing that happened recently? Yeah. Yeah, this is a very hot issue, obviously, and we're all very curious to see what's going to happen with all these things. I have a feeling that type of editing, where if you're going to have a baby, so let's say you're getting married. Here's where I think the first one is you're getting married, you love this person, and you want to have a baby. And then you go do some genetic testing, so you get both of your DNA sequenced, and you find that you're both carrying some mutation that once you have the child, 100% chance, there can be cases of this where your child will have some disease, or maybe 25% chance or 50% chance. I can definitely envision a world where you still have the baby and you ask them to please just correct that one mutation so that they won't die at an early age. I definitely could see that. In fact, that was what was supposed to be happening with this CRISPR babies. They were trying to do something similar to that. So I think that's a start. I think another area could be, let's say you do have some precancerous something on you, and let's say we can do the DNA sequencing and we see that there's some mutations that have happened on your skin, and this is going to end up most likely causing skin cancer in that region and might spread. I could see trying to do some kind of like local gene editing in a region of your body to try to kind of make them normal again so that they're not going to be cancerous anymore. I don't know exactly how that's going to work. It's not going to be me doing that particular part of it. I would say those are going to be the first big two, and of course the one that this is already happening. And the ethical piece of it, it's like, it's tough. The ethical piece. Yeah, I don't have like a clear answer on the ethics of that. I think if two parents, if they do enough studies and they try this out enough, like the baby, like let's say really early gene editing when you're having your baby, if they do it enough times, this is like, I heard a story recently about, I think it was in vitro fertilization. Apparently, when that idea came out, everybody thought this was going to be like the end of the world, in vitro fertilization. All the babies are going to be totally messed up, and it's not going to work, and it's unethical. And look at how it's changed on that today. So it could be similar to that. And the ethics piece, I think I'm not going to be the one who's going to answer that one.
Last question.
Okay.
You know what, I should pass this to him because that was my question, editing. So thank you for presenting.
Yeah.
Last question.
Okay. Could you image our DNA at an early age and then just maintain that into infinity?
Yeah, like without any mutations as your...
Yeah, you wouldn't have any diseases or cancer starting to form, just revert it back to the original image. As we get older, revert it back, fix whatever's causing us.
Yeah. Well, these are the things that keep me awake at night, I would say, honestly, because it's like I'm running out of time. I'm already aging myself, and I'm worried about it. so yeah, along those lines, it may not just be, everything I've talked about so far is like mutations, like one letter, let's say, changes to another letter. There are other things that cause you to die and cause you to age and things that's not necessarily just the mutations. That's one piece of it. For instance, cancer, that's going to play a major role. There are other ways that you can die, heart attacks and things like that. It doesn't matter if you change your base pairs or not. So there's only going to be like a subset of illnesses that we can tackle with this. And we're all still probably unfortunately going to die from all these other ones if we figure that piece out. But yeah, I do think there could be something there. I don't know how exactly you would do that, where like every single time your cells are replicating, you somehow go in and double check and make sure it did it correctly. It's probably a little too expensive, and that's probably not going to ever happen, I'm guessing. And then again, as far as like trying to revert it back, this is where I think this gene editing, maybe there would be some kind of local region that has a tumor forming or something, and you could maybe go in and edit out some of the changes to specific regions that influence the cancer to grow in certain ways, I think.
Good start.
Yeah. Yeah.
All right, let's give a round of applause to Leo. So yeah, I hope y'all found that as interesting as I did. I think it's really cool to have Leo come out here and give a talk about something that none of us really work with. Definitely out of our wheelhouse. So I wanted to just talk a little bit about the organization. So this whole thing was put on as an event for TECH, the Technical Employee Community of Houston. And some of the goals of this organization are to, engender more of a community within Houston specifically with technical related topics like this. And so we are planning to have more and more people like Leo, like Dr. Lienhard, come here and give talks and get us thinking and talking about technical topics that keep us curious and involved and innovative. I don't think I can get my slides up on your machine, unfortunately. But on these packets that I have put around the room, there are some QR codes that you can follow to get to our website and to let us know what kind of topics you might be interested in. And we will put them on our roster and try and find someone who can talk about that topic. Or if you have someone in mind in particular, then we can reach out to them and have them come. And in addition to just these events where someone is up here speaking at you, maybe we can have more interactive things like a hackathon or a round table or something like that. And yeah, so if you're interested in getting involved, there is another QR code on the back there where you can follow that, and it just like gives me your email address so I can then get in contact with you. But yeah, I'm really glad to see all of y'all out here. I hope you had a wonderful time. If you have any questions for me about tech, then I can answer those now. Or if not, then thank you very much.
It was going to chill around here a little bit.